Hello, everyone, and welcome to today's webinar. Our topic for today is Elucidating Metabolic Networks in the Tumor Microenvironment. Today's webinar is sponsored by Biolog and hosted by Jen. Thank you so much for joining us. I'm Uduak Thomas, Senior Editor with Jen, and I'm delighted to be your host for today's webinar. Our main speaker today is Dr. Kostas Lysiotis. He is the director of the graduate program in cancer biology at the University of Michigan. But before we get to his presentation, you'll hear a brief talk from Dr. Dina Finnan, a senior product manager at Biolog. Now, before we move on to today's presentations, let's go over some background. Pancreatic tumors are dynamic pseudo-organs that contain numerous cell types. A typical tumor is made up largely of stromal fibroblasts and immune cell populations rather than actual cancer cells. These non-malignant cells work together to create a dense fibrotic matrix that blocks cancer cells from accessing nutrients and oxygen. That leaves them to employ some unorthodox methods to get what they need to survive. Dr. Lysiotis and his team have been studying pancreatic tumors for some time. They've been looking especially into the autonomous reprogramming of metabolic processes in cancer cells that helps to facilitate their survival and growth under challenging circumstances. His presentation will cover new research into pathways of metabolic crosstalk that takes place among pancreatic cancer-associated fibroblasts, tumor macrophages, and malignant pancreatic cancer cells that work together to help maintain tumor growth, survival, and therapeutic resistance. During the presentation, he'll also talk about some of the tools and techniques that the team uses to explore the inner workings of metabolic networks. We're just moments away from getting things started, but a quick reminder that we'd love to take your questions following our main presentations today. So please enter your questions at any time in the Ask a Question panel on your screen. All right, let's go ahead and get things started. Dr. Finnan, over to you.
Thank you everyone for joining us today. I'm excited to kick off this webinar with just a few words about Biolog. I'd like to talk about some of the things we've been doing and the brand new instrument we just launched. So Biolog and our core technology have been around for a long time, nearly 40 years. We have instruments installed and being used all around the world on 6 continents, with over 5,000 publications from researchers citing the use of our kits and our products for so many different interesting applications. Our technology focuses on understanding the metabolism of cells and using that to characterize cells' phenotypes. So what different cells are able to survive or thrive on and how that affects their behavior. And this core technology can be used for many applications, anything from microbiological QC, environmental monitoring, to bioprocessing, where the goal is to improve the yield of a product you're trying to make using cells. And of course, it extends into all kinds of important basic research and discovery and development of new drugs. And so I'm sure you've heard the expression, you are what you eat. And I think that one of the best kind of multicellular representations of that is the flamingo. So baby flamingos are gray, as you can see in this cute picture. And as they eat the algae and the brine shrimp in their environment, their bodies metabolize those natural orange and red pigments that are found in their food called carotenoids. And those turn their feathers pink. And so, of course, this is the phenotype that makes flamingos what they're famous for, but it has nothing to do with genetics at all. And that's really what Biolog's technology focuses on at the core of it, looking at what cells can eat or metabolize and what that does to their behavior. And the food sources in this case, of course, are chemical substrates that are made of basic building blocks. And I'm sure you know that breaking these macromolecules down is tightly controlled by enzymes, and many of these enzymes need cofactors like NADPH to turn over their substrates. So we add a redox reporter dye that is cell permeable, and when it's oxidized, it's colorless. When it gets reduced during the cell's metabolic processes, it changes color irreversibly. And so we measure this color change to be able to answer all of these different questions. So what are cells eating? We can do this by putting them in a minimal sort of media and giving them access to only one substrate at a time in each well of a plate. We can also, answer how much of the food source they can eat by making quantitative measurements of the dye reduction. And then, of course, how fast they can do it. And this is because we're taking kinetic measurements over time to be able to get a lot of data about many substrates all at once. And so here I'm showing an example of one of our set of plates that we call phenotype microarray panels. And we have these comprehensive sets that are specifically designed for profiling different types of cells, whether it's bacteria, fungi, or mammalian cells. And you can probe many different pathways at the same time. So you can see that some of the substrates here are specific carbon sources, specific nitrogen sources. There are inhibitors, so you might have antibiotics or antifungal compounds. And then we have combinations of conditions, such as various amino acids at either low or high pH. And so this set of, in this case, 20 plates looks like a large, overwhelming set of plates, but it's really where our instrumentation from Biolog comes in to help. So this is actually a very quick and straightforward experimental setup because these plates come pre-filled with all of these different conditions. And so you just pipette the same cells into every well of the plates, you stick them in the instrument, and you just let it go. And because there's room for 50 plates, that means you can measure up to 4,800 conditions at the same time, which is a very powerful way to thoroughly characterize one cell type or make comparisons between different cell lines or cell types. And so here is the set of those phenotype microarrays that are specifically designed for probing energy metabolism pathways in mammalian cells, and which you will hear more about from Dr. Elucidating today. And you can see that there are specific panels with different sugars, amino acids, lipids, and peptides. And there's other panels with drugs and various metabolic effectors such as ions and hormones. And so this really streamlines the workflow for being able to screen through many conditions at once for mammalian cells. And I'm excited to introduce you very briefly to Odin, which is our newest platform for being able to incubate and read any Biolog plate. So with Odin, you can measure the formation of the redox dye, like I mentioned briefly, to look at cell metabolism. And you can also use more traditional optical density measurements to measure cell growth. So we read each plate at 2 wavelengths, either 490 or 590 nanometers and 740 nanometers. And because it holds 50 plates, it can handle any throughput. So from 1 plate at a time to 50 plates, and multiple users can run plates at the same time. And I'll end this section by showing that we have a whole ecosystem at Biolog to help our researchers characterize cells, whether they're microbial or mammalian. And we do this with our fit-for-purpose plates that are designed for specific applications, and our instrumentation. And so Odin, as I mentioned, is the brand new 50-plate version of the instrument that can do everything that our older instrument called the OmniLog that you'll hear about from Dr. Elucidating next. This can do everything that could do, plus more. And we've really modernized the software for Odin and made it even easier to acquire and analyze and export all of the rich data that you're collecting. And then our ecosystem also includes protocols, training, and support from our expert technical support team. And then finally, we have a lab service and testing facility that is ISO certified and can do all of this phenotypic characterization that I talked about today, as well as other lab services that are very relevant, particularly to folks working with microbes. And with that, thank you so much for your time today. I'm excited to pass it off to our main speaker to learn more about how his group is using this technology to better understand tumors and their microenvironments.
Hello, my virtual friends and colleagues. My name is Dr. Kostas Lychotis. I'm an associate professor of physiology and medicine at the University of Michigan in Ann Arbor. Our research group studies metabolism in cancer and immunity, and I'm excited to share a new story from our group concerning metabolic networks in the tumor microenvironment. I would like to start by thanking Biolog and the event host, Genetic Engineering and Biotechnology News for the opportunity to share new research from our lab. I would also like to thank those of you signing in virtually for your time. If you have any questions that extend beyond the Q&A session, please contact me by email or social media at the links provided here in the corner. As a matter of diligence, I have detailed my disclosures here. Of particular note, Biolog provided instrumentation for this study. However, they did not provide reagents for financial support, and Biolog had no influence on the outcome of this work. Today, my talk will focus on metabolism in cancer. As a primer, allow me to briefly introduce a few concepts in tumor biology. A helpful way to think about solid tumors is as pseudo-organs that form outside of normal developmental control. It is important to note that this includes a number of distinct cell types, like those I've listed and pictured here, and these interact inside of a tumor to create unique physiology. A consequence of growth outside of developmental control is the formation of leaky or insufficient vasculature, the formation of nutrient and oxygen gradients, and the accumulation of metabolic waste and disrupted pH, culminating in disrupted metabolic properties and programs. My lab has been interested in understanding how these metabolic disturbances are managed within a tumor and even harnessed to support the growth of the tumor. We have focused our attention on intratumoral metabolic crosstalk pathways. These we categorize as being cooperative in nature, like those pictured here. Competitive. This is often concerning ways that the tumor is combating or resisting the anti-tumor immune system, and those that mediate therapeutic resistance, and this can be to immune-based or other types of therapies. My lab studies these concepts in pancreatic cancer. This is a tumor type characterized by a low proportion of malignant cells, as is evidenced here, surrounded by this dashed line. And I trust you do not need to be a pathologist to appreciate that the proportion of malignant cells relative to everything else that is in this tumor is low. These tumors are extremely complex, again, as evidenced by this image. They also are marked by having low vascularity, which results in deregulated access to nutrients, since serum-derived nutrients are not readily available. And pancreatic tumors are considerably resistant to virtually every therapy we have in the arsenal. Targeted therapies do not exist, and immune therapies have yet to show any promise in this disease. That brings me to the major questions in our lab. How is metabolism rewired in the cells inside of these tumors to support their survival, growth, and resistance to therapies? What are the nutrients available to the pancreatic cancer cells? And if they don't have access to the vasculature, where are these nutrients coming from? How do cooperative metabolic crosstalk pathways exist in the TME? And what purposes do they serve? Are these the source of the nutrients I mentioned in the bullet point above? Do the deregulated nutrient profiles inside of a pancreatic tumor influence the way that the anti-tumor immune system recognizes, or in the case of pancreatic cancer, is unable to functionally recognize and attack the tumor? And as we're working through all of these sorts of big picture questions, we are mindful of recognizing opportunities for new drug targets and to develop new and more effective therapy. These mechanisms are perhaps best described by way of example, and I will use this opportunity to highlight work from our group. Cooperative interactions are those where one cell type, for example, cancer-associated fibroblasts or CAFs, provide nutrient support to a second, like the sharing of amino acids or even extracellular matrix that can be metabolized, like the sugar polymer hyaluronic acid. If you'd like further information on these examples, I'll point you to the references at bottom right. Competitive interactions are largely used to restrain the activity of the anti-tumor immune system. Like cancer cells, cytotoxic T cells also require nutrients to proliferate and mount effector responses. We have found that a major mechanism by which immune suppression is mediated is through amino acid degradation. And I provide again several examples here of recent work from our lab, and for further reading, point you again to the references at bottom right. And finally, are metabolic mechanisms that mediate resistance to therapies. So these could be immune therapies like the degradation of amino acids I mentioned just a second ago, or resistance to other chemotherapies as we have described for gemcitabine and mitochondrial poisoning. The story that I will share today concerns uridine metabolism, or uridinolysis. Uridine is pictured in the middle of the slide, and this term, uridinolysis, was made up by Alexis Jourdain and Vamsi Mutha, who co-published work on uridine metabolism alongside of our study. And so credit where credit is due, but we like the term, and so we're going to use it here. All right, the project I will tell you about today, I'd like to briefly summarize before we get into the data so that everyone has a sense for the direction and is better able to follow along. This project was co-led by a collaborative group started by Matt Ward, who is now a graduate student in Gary Patty's lab at WashU. It was picked up by Zuribe Nosu, who just recently started his own lab at Cornell in Ithaca. We got a bunch of support from a senior graduate student in the group, Pete Sedgwick-Nukit. And finally, this has been a long-standing collaboration with Pawan, and you'll hear about where each of these individuals contributed as I move through this presentation. Additionally, I encourage you, if you're into social media, to follow Zuribe, Sadnan, Dam Lab, and Pete on Twitter to keep track of their progress and accomplishments. All right, in short, in this story, our teams demonstrate that uridine is present in the tumor microenvironment, that it can serve as a novel metabolic fuel to feed cancer cells, and that this is able to support tumor growth when glucose is limiting. Now let's get into the story. This project started with a relatively simple question, and this is where Biolog came in. I mentioned during my introductory remarks that pancreatic tumors have restricted access to serum-derived nutrients, yet they grow. So we were interested in understanding that the compendium of nutrients that a pancreatic cancer cell could catabolize. So we teamed up with Biolog, and we used their platform here to assess the activity of nutrient utilization using an arrayed system, a kinetic monitoring device, and then we credentialized leads based on correlation with gene expression. All right, so this was a rather large screen, and I'm going to walk through it in 3 parts. I think it's also useful to understand as I walk through these our thinking behind how we design this screen. So we assessed the activity of around 175 metabolites. We did this in screening media. The substrates are in roughly millimolar concentration. They are lyophilized into an arrayed fashion on these 96-well plates. We used severely nutrient-restricted media, the purpose for which is to be able to identify our signal, our activity of a metabolite over baseline. As you all likely know, cancer cells in culture have been adapted to grow in glucose and glutamine over the course of, in many cases, decades. And so they use these exceedingly well, and inclusion into a screening assay would dampen the ability to observe meaningful hits. And so we utilized low concentration dialyzed serum so that it didn't include any metabolites in the assay. Those 175 metabolites, if you're curious, these are PM1 through 4 plates. Can be roughly broken down like this. There were a number of metabolic intermediates, carbohydrates, lipid intermediates, amino acids, nucleic acids, these are the ones I'm going to focus on, and so on and so forth. All right, the second aspect of the assay was to determine activity. We used a panel of established pancreatic cancer cell lines, so these are all commercially available cell lines, 20 of them, and we included 2, what I'm going to call normal, these are non-transformed cell lines. Our early intention was to include those to differentiate them from the pancreatic cancer cells. That, for this particular study, that ultimately didn't provide useful, but I think it was still a smart inclusion. So we have a large panel of cell lines to make sure that we're not looking at artifacts from a small number. And we plated these cells onto the lyophilized plates, and to measure activity, we took advantage of an old approach, in fact, that relies on the reduction of this blue rosazurin dye into a colored pinkish purple resirufin dye. And this is a reflection of cellular reducing potential in the form of NADH. So metabolically active cells are generating NADH through glycolysis or the TCA cycle, and that NADH is being created when nutrients are metabolized. And so as a reflection of metabolism, we were able to look at this conversion from blue to pink, pinkish purple. And then we monitored, this was not an endpoint assay, I think this added some real utility to it. By using this biolog instrument, effectively a large incubator with mounted camera plate analyzer, we were able to run these plates simultaneously. Maybe we had 4 or 5 batches, bearing in mind running these plates in replicate on 20 cell lines, okay, and we monitored the conversion, right, the NADH generation as a function of time. So every 15 minutes, one plate would move in front of the camera, an image would be captured, that plate would be put back in place, the next plate would be, and this would go on cycle, and we did that for 3 straight days. And so what you generate from that sort of assay are these curves, which provides an enormous amount of information. We can look at the lag phase. So how quickly does a cell, once plated, begin to catabolize the nutrient that is in the plate? How quickly is it able to do so? That would be the slope. What is the maximal activity? Where does that plateau? Or one can look at the area under the curve. For the purposes of our study, for this study, we utilized area under the curve, though each of these individual parameters provided an enormous wealth of information. And then the last bit, so we had a number of what I would call interesting hits, but how did we decide which one to pursue? So I'll give you a couple of examples here. Glycogen utilized by all the cell lines, pyruvate utilized by some cell lines to high degree, right, where relative metabolism or NADH production is on the y-axis, cell lines are on the x-axis, and you can see that there's variation in the ability to use pyruvate. Two very similar molecules here, ketobutyrate and hydroxybutyrate, again, among the cancer cell lines exhibit this sort of variation in utilization. But I highlight these two because here you have one molecule, I mean these isomeric species, one is well utilized by our normal cells and one was not, or unable to be utilized by our normal cells. So which of these molecules do we pursue? And this is where Pawan and the SED and ANDAM lab really came in. We said, let's take all of this information and correlate the activity with gene expression of the metabolic enzymes. And so this data set was bulk RNA-seq pulled from the CCLE, the Cell Line Cancer Cell Line Encyclopedia, the Broad Institute. And this is a busy slide, so let me briefly summarize here. What we've illustrated on top, this is how we identified our leads. These were the 12 best hits. In this purplish color, those are negative correlations between the expression of a metabolic enzyme across our panel of pancreatic cancer cells and its ability to be metabolized. In orange are positive correlations. So shown down here are, and these data are all in the paper, if there's interest in digging deeper. But you can see 2 negative correlations, 2 interesting negative correlations, and 2 positive correlations. We ultimately decided to pursue this particular positive correlation for a variety of reasons, but the very first of which is the metabolite uridine is in the name of this enzyme uridine phosphorylase. So that was the first indication that we wanted to go after, but I'm going to give you a little more data now to that really bolstered our confidence that this was interesting as we dug deeper and deeper into it. So in essence, that was the library. So now we have our hit. This is UPP1, the enzyme, and uridine, the metabolite. And what is the connection between these two across or in pancreatic cancer and in pancreatic cancer metabolism? So the first thing we did was reproduce our data outside of a screening platform with our own gene expression, so qPCR gene expression relative metabolic activity. The correlation we saw earlier held. In fact, it was a little bit tighter. You can see here these are a variety of cell lines from our panel, and then in base media in white, in orange is the addition of 1 millimolar uridine. And you can see well recapitulated that variation in activity. Every cell line that we observed was able to metabolize uridine among these pancreatic cancer cells, just to a varying degree. We thought that gave us a nice window to study. And I think it's also worth pointing out that the correlation here of UPP1 and relative metabolic activity was not true for any of the other nucleosides that were in this screening deck. So here's adenosine in red and inosine in green. You can see that those are actually well metabolized. They're very high on the relative metabolic activity scale, which means a lot of cells were able to use them for metabolism, but there was no correlation with UPP1. Same is true of thymidine. Thymidine down here was not metabolized, and nor did it correlate with UPP1. So we thought we had a specific correlation between uridine and UPP1. Next, we interrogated publicly available data sets, and these revealed that UPP1 expression predicted for worse outcomes in pancreatic cancer cohorts. This was evident in 3 of 4 cohorts that Zaribe analyzed. You can see the orange line. That is patients who had high expression of UPP1 relative to those in the black line had low expression. UPP1 expression was also correlated with mutant KRAS in the TCGA. Mutant KRAS is the signature transforming oncogene in pancreatic cancer, and the fact that the expression correlated with this important oncogene gave us further evidence that UPP1 may be important in pancreatic cancer and worth further pursuing. Finally, we made use of a publicly available data set, in fact, that we deposited some time ago when I was a postdoc, using the iKRAS mouse model of pancreatic cancer. With this model, we can feed the animals doxycycline, and this activates mutant KRAS specifically in the pancreas. And here, what we see is that when we turn on KRAS in pancreatic tumors, that UPP1 expression is elevated. What this particular experiment illustrates is that, as opposed to the data in the middle where it's correlative RAS and UPP1, this is direct evidence that RAS has some role over UPP1 expression. So these data in sum encouraged us to take a deeper look at uridine and UPP1. From the original screen, our data revealed that uridine could fuel metabolism in glucose-limited media. So at face value, this might seem like an exciting discovery, but I'll tell you, it's actually dates back even before I was born. There's studies I show here from the 80s and the aughts, but even papers in the 60s. And those studies illustrated that your cells could grow on uridine, and the thought has been the role of uridine in this capacity is to promote nucleotide biosynthesis. So in essence, uridine is utilized to fuel pyrimidine biosynthesis, and when the ribose is cleaved from uridine, that can be used to recharge other adenylates like guanylates. Adenylates and guanylates to support cellular biosynthesis. And our data were in line with this model. We found that UPP1 expression and activity correlated with the removal of ribose from uridine. That's illustrated here. When we feed the cells uridine and analyze uridine and uracil levels by mass spectrometry, we see massively elevated uridine. Unsurprisingly, we fed them that. But we also see an accumulation of uracil, a dramatic accumulation, hundreds of fold, in the extracellular and the intracellular space, really providing further credence for this idea that UPP1 is functioning as expected, removing uracil from the ribose base and providing ribose, making it available to the cells. This we repeated in multiple cell lines, as I illustrate here. But our screen was designed, in fact, to identify metabolites that supplied the cell with reducing potential. Here is NADH. And you can see that NADH is below nucleotide salvage, and therefore not accounted for by classical mechanisms. So we put forth that the uridine-derived ribose is used to fuel glycolysis and central carbon metabolism more generally by rerouting through the non-oxidative pentose phosphate pathway. And we observed in multiple cell lines, one's presented here, that uridine and glucose carbon fuel the same pathway. That is, as illustrated here, when you add uridine in orange to cells with glucose, there's no impact. That's contrasting to cells with no glucose and no glutamine. When you add uridine, you have more metabolic activity. And this is further increased when glutamine is added, indicating that uridine and glutamine are operating through distinct pathways, but uridine and glucose are operating through the same pathway, or providing further credence for this idea that the ribose was entering into glycolytic and central carbon metabolism. To further substantiate that point, we're able to recreate much of the activity that we observe using uridine by just adding extracellular ribose to the cell, right? And that was not something we observed with uracil, so this was just true of the ribose. I just have ribose pictured here. We then went to take a deeper dive in understanding how the uridine-derived ribose was utilized with isotope tracing and metabolomics. And in this case, we see that uridine-derived ribose carbon. So we've used non-radioactive heavy isotope labels to put C13 on each of these ribose. And we can trace that using mass spectrometry and metabolomics. And what we found is that when you feed cells this uridine with the labeled ribose, we completely turned over the uridine pool. We saw expected incorporation of uridine into UMP and UTP, so that's through the pyrimidines. But I won't walk through each of these individually, just suffice it to say, if the bars are colorful, that means those isotopologues those are species that contain the heavy isotope here. So let me zoom in on one example. Here's citrate, right? So citrate branches off from glucose metabolism by way of pyruvate entry into the TCA cycle. When citrate is made from glucose, or in this case, uridine-derived, ribose-derived carbon, those heavy atoms will end up on the citrate, and that is exactly what we see. There's a variety of isotopologues. I don't need to walk through each of these. We see enrichment in glycolysis. We see enrichment in a number of branch pathways. We see enrichment in the TCA cycle. So this was really exciting, and it was truly reflective that uridine-derived ribose could indeed be used to fuel metabolism. For the metabolism aficionados, I'll say, given time constraints, all of this information is in the published study, and I encourage you to take a look at it. Furthermore, when we also wanted to validate that this could occur in vivo, and we saw, in fact, near identical patterns of uridine metabolism in vivo to those we observed in vitro when we delivered isotopically labeled uridine to mice bearing either orthotopic or subcutaneous pancreatic allografts. So these are immune-competent tumor models. We delivered the uridine by IP injection, and we sacrificed the animals and looked at uridine incorporation 1 hour later, and even in that short time frame, you can see the uridine incorporating into to ribose, that's through the pentose phosphate pathway, and through glycolysis, lactate metabolism, and into the TCA cycle, and demonstrating proof of principle, uridine is a metabolic fuel that can be utilized in vivo. Our previous work relied on superphysiological concentrations of uridine, and in order to study this more accurately, what we then wanted to do was quantitate the amount of glucose in uridine in pancreatic tumors, apply those concentrations in our in vitro systems, and then analyze how uridine was catabolized. So we teamed up with Alex Muir's group. We found that who's really pioneered this technique, at least in the last 5 or 10 years. We found that glucose concentrations in the pancreatic tumor interstitial fluid were in the low millimolar. Uridine concentrations were in the middle micromolar range, so somewhere between 25 and 50 micromolar. It was enriched relative to the plasma. We similarly saw that uracil concentrations were higher in the TIF than in the plasma. In the interest of time, I won't belabor this study either, but suffice it to say, when applying in vivo relevant concentrations in vitro, we saw that uridine-derived ribose was still incorporated into central carbon metabolism quite robustly, and that this was impacted by the concentration of glucose. So we believe that the two are competing. The higher the glucose concentration, the less uridine was incorporated, and the higher the uridine concentration, the less glucose carbon was incorporated. So there's really a competition between these two, reflecting that under circumstances where glucose concentrations are low, uridine can be utilized as a metabolic fuel. And again, for the metabolism aficionados in the lab, if you want to look through these data in greater detail, I'll point you to the published work. So we've talked a lot. Now we started with UPP1 and we talked a lot about uridine, but let's get back to UPP1. So to illustrate a bona fide connection between this metabolite and this enzyme, we use CRISPR-Cas to knock UPP1 out of here. Pictured two human cell lines. These are independent clones from single cell clones from each of those cell lines. You can see that the relative metabolic activity, or NADH levels, are not rescued when uridine is added in the UPP1 knockout, comfortingly, sort of as expected based on our mechanism. And if we use mass spec to determine the levels of uridine and uracil, you can similarly see that uridine accumulates and it is not catabolized into uracil. Really nailing UPP1, we think, to the major mechanism by which uridine-derived ribose is liberated. Okay, we again used isotopically labeled uridine-derived ribose. We fed that to cells. This is in culture, in our wild type, in our UPP1 knockouts. I'm going to just jump to the next slide here. And again, I'm not going to belabor this in the interest of time. All of these data are available in the paper. And even if you don't love looking at isotopolog diagrams and distributions like we do, I trust you can still appreciate that the wild type cells, shown on the far left, as indicated by the colorfulness, are able to incorporate ribose-derived uridine into central carbon metabolism. This is what we showed earlier. Here now we've knocked out UPP1, and you see that the ability to put ribose into central carbon metabolism is completely lost. It's not there at all. It's not evident. That was really exciting. So we know that UPP1 is the major mechanism by which this is happening in pancreatic cancer. So how is UPP1 regulated? We looked at its regulation. I'll start by saying when I was a postdoc with Hao-Chung Ying, now running a lab at MD Anderson, and Alec Kimmelman, now department chair of rad-onc at NYU, we found that mutant KRAS regulated anabolic glucose metabolism through the MAP kinase pathway by regulating glucose uptake and glycolysis. So we add to this now with our new work to show that UPP1 is similarly regulated by mutant KRAS downstream of MEK and ERK. We demonstrate here using mouse models where we turn on mutant KRAS, and you see that UPP1 expression on the y-axis goes up. These are two independent cell line models. And reciprocally in human cells, if we inhibit MAP kinase with trametinib, you can see that here's plus trametinib, UPP1 expression plummets. Furthermore, we believe that uridine lysis is augmented under nutrient deprivation. So MAP kinase is the primary driver of UPP1, but under low glucose or low uridine, that can be further modulated. This is a complicated graph. UPP1 expression is shown, though I haven't written it here, on the y-axis. These are the conditions shown on the x-axis. Right? So in the far right, that is no glucose, no uridine, no trametinib. So what you have is MAP kinase on and no nutrients. UPP1 is the highest. If you add trametinib, inhibit MAP kinase, that comes way down. If you compare this bar where you have no nutrients, no trametinib, to full nutrients, no trametinib, you can see the comparison between these two. Again, comes down. So we think of the nutrient availability as the dial knob, the volume knob, further governing UPP1 expression. And we think this also makes intuitive sense. If the cell is energy starved, it's going to turn on UPP1 to start chewing up its uridine to make energy. All right, so we validated everything we did in this paper. We did in multiple cell lines to convince ourselves that this was indeed real. We found a number of features that were cell line specific, but those which were generalizable, we presented as conclusions in this paper. And so, if you're interested, I encourage you to check out the study. All right, last, potentially but not least here, we now looked at the ability of UPP1 knockout and its impact on pancreatic tumor growth. In this case, we teamed up with the Pasca di Migliano lab here at the University of Michigan. They have a number of really powerful mouse models of pancreatic cancer. In this case, we used a tumor allograft model. And what we do is we take syngeneic pancreatic cancer cell lines. So those are genetically matched to black 6 animals. We put those cells into the pancreas of these genetically identical counterparts. And the reason for that, number one, is the animals have an intact immune system, so we can study the impact of tumor immunity. And second, we put the tumors in the pancreas because that's a more physiologically relevant microenvironment to study tumor growth. So vector control shown at left, tumor weight on the y-axis, you can see with two independent knockouts of UPP1. The tumors are considerably smaller, and this we did with 2 independent model systems. I'll point out we validated the mouse cell lines for metabolic activity using the same panel of assays we used for the human cells, but in the interest of time I didn't show that. As a marker of PD, we also show that in these tumors uridine accumulates and uracil decreases. So 7940B model, it's the one here, and the MT3 model far right, and then that corresponds to the far left. We also noticed what I think is a really interesting observation in that the decrease in vivo growth was remarkable when compared to the in vitro growth of the corresponding cell lines. So these cell lines in full media, this is DMEM, 25 millimolar glucose, no uridine in that media except the little bit that comes from the serum. These cells grow just fine when you knock out UPP1. No observable defect. So the vector controls in salmon, you can see it either sits right on top of or even below slightly below this one knockout here, or they're just all on top of each other there, reflecting that when glucose in particular is not limited, there appears not to be a necessity for UPP1. But under circumstances where glucose is limited, perhaps that's when UPP1 becomes more relevant. I will mention another stark difference between these systems is the lack of a tumor microenvironment. So there's still a lot to learn here, but I guess I'm pleased to see that we have a really great model system to begin teasing apart the nutrients that are in vitro and then studying how that impacts their growth. All right, so that brings me to the conclusion. We found that uridine is a novel metabolic fuel in pancreatic cancer whose metabolism is mediated by UPP1. UPP1 expression is regulated by KRAS MAP kinase signaling, and this is augmented by nutrient availability. Finally, we show that UPP1 inhibition is growth inhibitory in immunocompetent orthotopic pancreatic tumor models. That's the last point here. With that, I would like to thank my awesome team, those who led this work. I mentioned earlier, but I'll remind you that it's Matt, Zaribe, Juan, and Pete. They were co-first authors on this study. Each of them provided absolutely important, non-redundant effort to this great collaborative study. I want to thank our network of really awesome collaborators, those who participated in this particular study. We got a big network for this study are bolded here. Thank our funding sources. Also like to extend my sincere thanks for those who paid attention to this talk. And I look forward to answering any outstanding questions during the Q&A. Thanks again.
And a very warm welcome to you all. Welcome to the live Q&A for today's webinar. We're so excited you're here, and we hope you enjoyed the presentations. Both of our speakers are here, and they're ready to answer your questions. So if you haven't already done so, please do submit your questions now, and we'll do our best to answer them live during this session. So thank you once again for being here, and welcome to both of our speakers. I would actually like to start off with you, Dr. Lysiotis. Is this unique to uridine, or are there other nucleosides similarly that are able to provide cells with ribose to support bioenergetics?
Yeah, that's really a fantastic question. Thanks for the question. Thanks for the opportunity to share our work. We found a couple other nucleosides from the screen that were actively metabolized, and I just touched on this very briefly. What we were looking for were correlations between nucleoside catabolism and gene expression. So the other nucleosides that were catabolized and that were included in the screen, adenosine and inosine, highly metabolized but not correlated with any genes in particular. So that's why we decided to go after uridine initially, but I think there's really a lot more action here. A number of other labs have begun to discover roles for nucleoside metabolism in other contexts. So didn't come out of our screen, but definitely an area still worth pursuing.
Absolutely. And I'm actually going to stay with you for this next question. What role, if any, do you think the uracil plays?
To be determined, but we think that this is going to be a super exciting area to pursue as well. We didn't dig into this for a variety of reasons. In vitro, we didn't see an effect from uracil, but I think there's good reason to believe that it may have a signaling role, and that signaling role is going to require a tumor microenvironment. So what's uracil doing? Nothing in cell culture, but we think that in vivo, it's interacting with different immune cells and maybe having phenotypic effects, but that's ongoing work happening in our lab and others. So stay tuned.
Excellent. And Dr. Finnan, I'm going to come to you right now. What are the main differences between the new Odin instrument that you mentioned in your presentation and the OmniLog instrument? If you could speak to that, please.
Yeah, sure. So, they're similar in some ways. They can both incubate up to 50 plates and, read them and provide all the data, but we've really modernized the new Odin instrument. So OmniLog that you heard about today, it used to take an image of each plate. It had like a CCD camera-based detection, and we've replaced that with a 3 different wavelengths for measuring optical density. So you can measure the dyes that we're looking at the reduction of, as I mentioned at the beginning, and you can also look at more classical kind of turbidity measurements because we are measuring optical density. So those are the main differences. And then, of course, we really modernized the software and the analysis and all of those capabilities.
Okay. And I think this question might also be for you as well. Do you have the list of compounds in the plates readily available?
Yes, we have plate maps. We are happy to share them with you. Yeah, just reach out to the email address that was listed, info@biolog.com, and we can share the plate maps with anybody who's interested.
Wonderful. And so for the next question, could primary cells be used in this method and not just cell lines?
Yeah, I can take this one too. Yeah, sure. Yes, we have really nice examples in the literature of people working with primary cell lines from patients. So yeah, any cells can go in as input into this assay. Yeah.
Wonderful. Thank you. And just a reminder to everyone, if you have a question, please do drop it in the chat. We're still taking questions, so please keep them coming. So for this next question, are there any other enzymes that can do uridinolysis, specifically phospholipase D? Can that do the same job as UPP? And I think this one might be for you, Dr. Lysiotis.
Definitely, yeah. I'll field this one, and then I'm looking through the list here, and if you don't mind, I'm going to field a second one too.
Absolutely.
Which is there's a pair of them that ask effectively the same question. Okay. So, can other enzymes do uridinolysis? We landed on UPP1 based on the correlation of metabolic activity to uridine and gene expression. We did not see any other enzymes with putative ribose extraction functions coming out of that correlation. Now, we only worked in pancreatic cancer cells, and to be honest with you, I don't know what the expression of phospholipase D is in pancreatic cancer. So if it's not expressed, then potentially. But if it is expressed, I would say the answer is no, and that's based on another piece of evidence, which is we knocked out UPP1 and we saw that ribose extraction from uridine was 0. And we did that with metabolomics and isotope tracing. So I think we can conclusively say in pancreatic cancer that the primary enzyme doing this function is UPP1. UPP1 has a sister homolog called UPP2. We found that was not expressed in pancreatic cancer, but it is important for doing the same job in the liver, for example. All right, I'm going to jump to this, Uduak, if you don't mind. I'm taking the reins a little here.
Yes, please, go ahead.
Yeah, 2 great questions here. The first says, what do you think is the source of uridine? Does it come from circulation or other cells? And then the second question says, which cell type is the main source of uridine in the TME? Love this question. Right at the heart of the research program going on in the group right now. We had initially speculated that the main source of uridine was tumor-associated macrophages. These are a type of immune cell that lives inside of a tumor. It's a macrophage, and because of its proximity to cancer cells, we call them tumor-associated. We had previous data illustrating that tumor-associated macrophages abundantly release pyrimidine nucleosides. So we said, that's got to be it. And we did that experiment. We knocked out the macrophages from the tumors. There are a variety of clever ways to do this. We ended up doing it with an antibody and pharmacological inhibitor. And so this data is detailed in the paper. And for those interested, I encourage you to look at it. The long of the short with that experiment is that bringing down the macrophages in the tumor had 0 impact on tumor uridine levels. and tumor uracil levels. It had a really big impact on circulating uridine, but that's a tangent for a different day. So, at least from that experiment, we don't think that macrophages are the main source. We did another experiment where we injected uridine IP into the animals, and I showed some of that data. You can see that the uridine enters into the tumor rapidly. Within an hour, we saw considerable labeling of the tumor from the uridine-derived ribose. So some of the uridine is undoubtedly coming from circulation. We couldn't account for all of it. I think another provocative model is that the uridine is actually cell autonomous. So think about turning over RNA when a cell is hungry in a manner akin to turning over glycogen when a cell is hungry, right? So glycogen releases glucose, RNA would release uridine. We're building on that model. It's speculation now, but I think it's quite compelling. But yeah, great questions. So that's where we're at.
Wonderful. Thank you for speaking to both of those questions. And just a reminder again, we have a couple more minutes to take a few more questions, so get those last minute ones in. Let's take this one. Do you think that pathophysiological conditions where uridine may be increased could promote cancer? And I think Dr. Elucidating, this one's for you too.
Yeah, thank you. I think that is a hypothesis worth testing. So my opinion, I think it could facilitate, not necessarily promote, right? It's one piece of a puzzle. And so I love the idea. And the way we're getting at that is we've just made the conditional knockout animals where we can make tumors in the pancreas of mice with genetic engineering and knock out UPP1. So what that'll tell us is if, I guess it partly answers the question, if the cell autonomous ability to break down uridine into ribose has an impact. And so if it is indeed the uridine-derived ribose, I think we'll be able to figure it out. But we have ideas like that too by manipulating dietary uridine. So I love the question. Again, just stay tuned for more.
Wonderful, wonderful. All right, the next question we have in here is, what happens if you force PC models towards B-FAO oxidation while limiting simple carbohydrates and lower protein nitrogen availability?
All right, so I think this one's for me, right? Yeah.
Yes, I believe so.
Everyone's comfortable with that?
Yeah.
Good. PC being pancreatic cancer and FAO being fatty acid oxidation. I think what this question is asking simply is what happens if you put an organism with pancreatic cancer on a ketogenic diet? And there's a bunch of great data to explain what happens now. So a ketogenic diet in isolation, I think the consensus in mice and man is it does nothing. But what it does is it changes the circumstances. And when the circumstances are changing, that can open up metabolic vulnerabilities. And Lou Cantley's group published a paper in Nature, maybe 2019 or so. Ben Hopkins is the lead author, where on a ketogenic diet, there's less circulating insulin. And what that does is make pancreatic tumors susceptible to inhibitors of PI3 kinase. That's quite a mouthful, but for the question asker, I encourage you to go check out that paper from the Cantley lab. I'm sure you will appreciate it. And then there's another recent study from the Rabinowitz lab where they showed that a ketogenic diet alone, again, does nothing, but in combination with, I can't remember what chemotherapy they did, but a pancreatic chemotherapy, they saw increased efficacy of the chemotherapy and some durable responses in immune-based models. So I think there is utility for promoting fatty acid oxidation or getting tumors to live on ketone bodies, just not alone. Think of treating diet as medicine, basically.
Wonderful. All right, we have another question about uridine. What are the main transporters for uridine?
Yeah, that's a great one. There are 6, if memory serves, transporters that can transport uridine, 3 concentrative and 3 equilibrative. In pancreatic cancer, I think of those 6 in our models, human and mouse, we saw 3 of them expressed, 3 of those 6, maybe it was 4. I don't remember which ones they were exactly. If you shoot me an email, if you're really curious, I can get you that information. Undoubtedly, we didn't pursue it because, I mentioned this very briefly, but I'll bring it up now. Vamsi Mutha's group published their paper on similar concepts in under normal physiological processes. So in essence, they found that uridine could be catabolized by normal macrophages and normal liver cells, and that has important governance of metabolic homeostasis in a healthy organism. But they ran some screens looking for dependencies of uridine metabolism. They also found UPP1. That's how they got to UPP1. They did not find any of those transporters. And so why am I telling you all this? Because I think there's redundancy in the transport network. So you knock out one of the other 6 will just take over.
Wonderful. I think I have another question that I may have skipped over earlier, so I'm going to go ahead and ask that now. This is asking about the first slide that you presented. If you could explain again how the uracil-derived ribose supports metabolism.
Okay. It's not the first slide, that's the punchline of the story, but happy to go over that. So uridine is a nucleoside. It's got a base and it's got a sugar, right? UPP1 breaks these apart. The base, uridine, now uracil, goes out into the trash can. The sugar, ribose, now ribose phosphate, enters into glycolysis through the non-oxidative pentose phosphate pathway. And then that can be treated equivalently like any glucose-derived molecule and enters into downstream glycolysis, TCA cycle, branch pathways, et cetera, et cetera. For more information on that, I encourage you to check out the paper. Check out the paper from the Mutha lab too, and Alexi Jourdain.
Wonderful. Thank you so much. I believe that is all the time we have for today's webinar. Thank you both of our speakers. We appreciate you for being here. And for our wonderful audience, just a quick reminder that the presentation and the Q&A session will be available on demand shortly for repeat viewing or to share with your colleagues. Please keep an eye out in your email. We'll be sending you that link that way. Once again, many thanks to Dr. Lysiotis and Dr. Finnan for being here and for their wonderful presentations. I'd also like to say a special thank you to our sponsor, Biolog. Thank you for sponsoring today's webinar. And I would also like to thank the superb Gen Multimedia Group for hosting. And that team is led by Liana Jabs and David Mosley, so shout out to them. And thank you to our wonderful audience for being here. We appreciate you taking time out of the day to be a part of this. Thank you for watching. And for everyone at Gen, I'm Uduak Thomas. Goodbye for now.